Hello, BISC130. This is the beginning of recorded lecture 4.5. Continuing on with evolution, we got through a lot of important stuff in the last one, but believe it or not, there is more to evolution that we're going to do in this lecture. So let's start by talking about something called the Hardy-Weinberg Principle of Equilibrium. It's quite a wordy title here. Let's look at Hardy-Weinberg. Oh, okay, so this is Hardy-Weinberg. It's a lot of math. Lucky for you, and for me, and for all of us, if you take an upper level, ecology, evolutionary biology class, you will certainly learn to do all of this. It's actually not that complicated, but for an intro class, let me try to put this into non-math terms. Hardy-Weinberg, or the Hardy-Weinberg Principle of Equilibrium, can be stated as the following. Allele and genotype frequencies in a population will remain constant from generation to generation. In other words, we're not going to see changes from generation to generation in the absence of evolutionary influences. Let's read this again. So, allele and genotype frequencies will not change over time if there are no evolutionary influences at work. This is a no-duh statement, right? So everything we talked about last time in the previous recorded lecture was about how all these evolutionary influences, whether it's natural selection or whether it's the founder effect or gene flow, that evolutionary influences cause changes in allele and genotype frequencies from generation to generation. Well, this is just kind of saying the opposite of that. It's you're not going to see changes if there aren't any of these evolutionary influences, if there is no natural selection, if there is no gene flow, whatever. Another way to think about this, and a much more common way to see this, is that if these changes are observed, and again, that's where all the math comes in about counting, population numbers in allele frequencies, stuff like that. If changes are observed within populations, then that means there must be evolutionary forces at work, and that's what the people analyzing these numbers then look for. If they're seeing changes in the allele frequency in a population from generation to generation, that must mean there are evolutionary influences at work, and then the job is, what are they? What is influencing the evolution of these populations? So, yeah, a very, very wordy description here, but in light of everything we talked about last time, this is kind of a no-duh. It's just kind of stating the obvious. If there are no forces, there will be no changes. If there are changes, there must be forces making these changes happen. Quick summary there. Now, let's look at how evolution shapes overall populations. So there are a few ways that this can happen, summarized here, but let's go through each of these one at a time to see what these graphs are actually looking at. So let's start with something called stabilizing selection. So stabilizing selection is defined in the key terms as selection that favors average phenotypes within a spectrum of existing variation. So you have variation from one end of a spectrum to the other end of the spectrum. The selection here is saying average is good. This side of things is bad, this side of things is bad. Whatever this phenotype is, the average phenotype is best and is going to be selected for. We need an example. Birth weight in humans. So birth weight in humans, it is a spectrum. Pay attention to the units down here. Yep, babies can be born, 3-4 pounds, all the way up to 11-12 pounds. As it turns out, the low end of this spectrum is bad. Low birth weight babies are, more vulnerable to complications is how this is phrased, and that's putting it pretty well. It's not great for babies to be born on the low end of things. That is selected against. Same thing with high birth weights. It is also more likely to cause complications as they're trying to come out, or to, the person that's been carrying this baby can have complications as well. So the high birth weight is also bad. It is this average that is selected for, and as a population, we have evolved to mostly give birth to babies in this sort of average zone here. These extremes still happen, but this is by far the most common one because of evolution. So it's shaped this population. The average is selected for. That's stabilizing selection. Next one, directional selection. So this one's also defined in the key terms. Let me read it as we look at this graph. Directional selection is selection that favors phenotypes at one end of the spectrum of existing variation. So yeah, you have an original population, whatever this selection is, it's saying the average isn't good, but this one extreme is good. One end of the spectrum is what is selected for. Example, javelinas, cactus. So yeah, these things they're related to pigs. They're not the same thing as pigs. They're sort of the American native version of pig. And yeah, they like to eat cacti. Cacti have some bristly spines on them. Most animals don't like to munch on them, but they could chew through these spines. They don't like them, but they could eat the cactus despite the spines. However, if the cactus has a lot of spines, it's not going to be worth it for these javelinas. They're going to tend to ignore cacti that have a high density of spines. It's just too much of a pain to deal with, and they'll go after cacti with fewer spines. So yeah, there's an existing variation within the cacti population of just how many spines you have. If we have this selective pressure at work here, it's specifically eating the cacti that have fewer spines and avoiding the ones that have more. That phenotype of having a high density of spines is going to be selected for. It's going to be a good thing. It's going to become more common. So yeah, this extreme of having a high density of spines is going to be selected for. That's going to shape the population overall to have that kind of phenotype. And the third one, diversifying selection. So again, I'll read the key terms while we stare at this graph. Diversifying selection is selection that favors two or more distinct phenotypes within a spectrum of existing variation. Most commonly, what this means is that there are two correct answers, usually the two ends of the spectrum. The average is not great. There's more than one right answer. Either extreme is good. Let's look at an example of this. This is a black-bellied firefinch, but there are many birds that experience this. There's a spectrum of variation in just how big their beaks are. As it turns out, small beaks are pretty good at, picking up small seeds. Big beaks are, they're not great at handling small seeds, but big beaks are great at cracking open tough, larger seeds. If there was a middle beak, the middle beak would be bad at both. The middle beak would be a little too clumsy to pick up and handle these small seeds, but a medium-sized beak would not be strong enough to crack open the larger seeds. That's going to be selected against these two extremes. Again, two correct answers here are both going to be selected for. That's what diversifying selection is. Two or more distinct phenotypes that are favored. So again, all three of these just examples of how populations have specific shapes with regards to phenotypes in the population as a result of the selective pressures that are on them. Now, so far all the evolution that we've talked about has been very small, changes in, beak size to accommodate the food in the environment, or, coat color in those squirrels in the recorded lecture last time. But we have a term for this when we're talking about smaller changes. The term for this is microevolution, which, if I can read the key terms, microevolution is changes in a population's genetic structure. So it's not always very dramatic. It's not always even anything that you can see. Sometimes it's just the genotype of things. But yeah, microevolution is just changes in the genetic frequency. The term macroevolution refers to larger scale changes. The key term's definition of macroevolution is broader scale evolutionary changes that scientists see over paleontological time. The thing that I want to emphasize is that both microevolution and macroevolution are the same thing in that they are both evolution. These are both changes in genetic makeup of populations over time. The only difference between microevolution and macroevolution is one of how long it takes, how big of these changes we're talking about. It's a sense of time. So here is analogy time. This is erosion. You got a drain pipe down here, looks like it's hitting this concrete, and over a couple of years it's wearing down this concrete so you can see sort of the pebbles underneath it. This is due to erosion. The Grand Canyon is also due to erosion. The only difference between this erosion and this erosion is, this took maybe a decade, this took millions of years. It took many years. We don't call this microerosion and macroerosion. They're both examples of the same phenomenon. In the same way that, we have microevolution and macroevolution. These are technically separate terms, but really they're everything that we've talked about so far. They're natural selection and genetic drift and gene flow. These two terms just are used to refer to, are we looking at small time scale changes, or are we looking at how changes happen going back a really long period of time? So we've kind of talked about macroevolution so far in these recorded lectures. We're going to turn our attention now to the bigger picture things, bigger changes. So as we go into this, we're going to be looking at speciation and new species and creating like really new things, not just slightly different colored squirrels. So before we head into that, I think we need to define what a species actually is. And this is actually trickier than it seems. There are many different ways to define what a species is. I'm going with the following one. So the definition that I'm using for this class is a species is a group of populations that are capable of interbreeding and producing fertile offspring. So interbreeding is sort of part of the definition. This does not work at all when you're talking about bacteria or things that don't breed, but for practical purposes, this is a pretty good one. So if they could breed and have good offspring together, viable offspring together, we consider them the same species. If they can't, different species. Example, dog, specifically poodle. Dog, specifically cocker spaniel. Dog, cockapoo. It looks very different than the parents. These two parents look very different from one another, but despite their phenotypic differences, these two can have fertile, viable offspring together. We consider them the same species, dog. Here's another example, donkey. Horse. They've got a lot of similarities, but they are pretty different in many ways. They actually can have offspring together, producing something called a mule. But the mule is not fertile. It's considered a hybrid. It can't have offspring of its own. And so donkey and horse, they can have offspring, but not fertile, viable offspring. Therefore, donkey and horse are considered to be different species from one another. And third example, bald eagle. African fish eagle. Very similar lifestyles, very similar diet, very similar coloration in the body and the wings. They can't have offspring together at all, despite their similar physical appearances. These are not the same species at all. So this, again, there are other ways to define species, but based on, having offspring together, that's a convenient way to define this. So now we're looking into speciation, forming new species. So speciation is the process of the formation of a new species, something different, something new. So in order for this to happen, populations of the same species need to change, need to evolve until they're so different from one another they can no longer interbreed. Again, if we're using interbreeding to define what a species is and what a different species is, we get new stuff by evolving separate populations until they're so different they can't interbreed anymore. There are two mechanisms for making new species. One of these is called allopatric speciation. Let me read the key terms definition as we look at this figure. Allopatric speciation is defined as speciation that occurs via geographic separation. So during allopatric speciation, we have starting population, something happens, some sort of physical separation occurs. Maybe this is, a volcanic eruption splits the island in two, and now they can't cross from one side of the island to the other. Or maybe a giant earthquake, rips open a chasm here, and, these small little lizards can't jump from one side to the other. Whatever it is, there is some geographic barrier that separates these two populations. And at this point, these are the same species. They're two separate populations, but if you were to grab one individual from this side and move it over here, they would be able to have fertile, viable offspring. That's why they're both green. That's what that is meant to indicate. But if you leave them geographically separated for a long enough time, and they're not doing gene flow, they're not exchanging ideas, they're not exchanging genetic information with one another, if you allow them to be isolated for long enough, they are going to accumulate differences. They're going to experience genetic drift differently. They're going to get mutations differently. And so these differences can accumulate so that over a long enough period of time, they have become different species. They've just changed from one another. They've drifted apart by so much to where even if you removed the barrier, or if you grabbed one of these lizards and plopped it over here, they would be different enough to where they could no longer interbreed, and we would consider them to be different species. To summarize that, yep, there's a geographic separation. That means there's reproductive isolation, no gene flow. They are separated from one another. And again, this can be due to a physical change in the environment. Again, some sort of geographical or geological thing changing the world around them. Or, and I'll show you an example of this in just a second, this can be due to dispersal of members to a new area. But again, when this happens, there's reproductive isolation. They've been physically separated from one another. And yeah, these two populations, they're just doing all that coin flipping, especially mutations are random, genetic drift is random. They're doing these random things independently of one another. It's going to lead to differences that over a long enough period of time is going to result in speciation. Example time. So let's look at one of these geographical, one of these physical changes in the environment. So the Caribbean and the Pacific Ocean used to flow freely into one another until about 3.5 million years ago. Due to tectonic plate movement, the Isthmus of Panama arose and separated these two bodies of water. This is not a very long strip of land, but I mean it's more land than a fish can cross, and so this effectively cut off these two bodies of water from one another. The fish on this side and the fish on this side are not experiencing gene flow, they're not exchanging information, and so they're going through all their mutations and genetic drift and stuff independently of one another. And again, this is 3.5 million years ago. That's a long time to do random coin flips and mutations and stuff like that. If you look at the fish on either side of this isthmus today, you've got a species of porkfish on one side, a species of porkfish on the other side. They look fairly similar. They're both medium-sized fish, but yeah, their genetic differences. If you look at their genomes and if you try to mate them with one another, they have been isolated for long enough. They're no longer reproductively reproductively compatible with one another, we consider these to be different species. And again, it occurred because of this physical separation, this physical change in the environment. Another physical change in the environment is actually, in my example from up here, the Grand Canyon. So it took a long time to form, but once it has formed, this is a pretty effective physical barrier between the critters on one side and the critters on the other. The birds don't care. You get the same species of bird on either side of the canyon, but for these ground squirrels, again, they look pretty similar to the eye, but if you look at the genotypic differences and if you try to have offspring between these two, they've experienced so much drift and so many coin flips separately from one another. These are different species from one another on the south side versus the north side. And I did make mention this could be physical change in the environment. Allopatric speciation can also be due to dispersal of members to a new area. So here's an example of that. You've got the Mexican spotted owl and the northern spotted owl. So, I guess maybe there was a rough year down here for these populations of Mexican spotted owls, and they were so desperate they just flew everywhere they could, trying to find a new home and new resources and new forests and prey. And any of those owls that flew out here didn't make it, and any of the owls that flew out here couldn't make it. But some of those owls flew over here to the west coast and found a nice place to live. They started a new population. They started to spread up the west coast. They're separated enough from the Mexican spotted owl to where, again, there's no gene flow. They're not hopping back and forth on the weekends to exchange genetic information. They are separated from one another. I guess this would also be an example of the founder effect if we wanted to analyze that. But the important thing here is that these two populations have been reproductively isolated for a very, very long time, and they've experienced, again, these genetic drift and mutations separately from one another. They've accumulated enough differences. Now they're different species: northern spotted owl, Mexican spotted owl. That's allopatric speciation. So that was my example: dispersal to a new area. The important thing is that there's a physical separation. There's reproductive isolation. This is in stark contrast to the other example of the other mechanism of speciation, called sympatric speciation. Let me read the key terms definition as we stare at this. Sympatric speciation is defined as speciation that occurs in the same geographic space. So there is no physical barrier. There is no reproductive isolation. You just have a single location, and within that, a new species arises. So, how does this happen? Well, we need to take a bit of a step back and look at selective pressures once again. Remember, selective pressures were any force that chooses who lives and who dies, what's good and what's not good, what is going to cause you to survive and reproduce or not survive and reproduce. Food sources are a very common example of a selective pressure. You need to get food in order to survive. You need to survive in order to reproduce. But the thing about food is that there are usually lots of different foods out there. And there are lots of different adaptations that help you get those different food sources. So this is an example of there not being one correct answer. There are multiple different adaptations that can arise to make use of these many different food sources that are available. So here's an example. These are cichlids, fish that exist within Lake Victoria, but a couple of other lakes in the world as well. And from a single sort of ancestral fish that colonized these lakes, we see speciation going in a lot of different directions. Again, they're all in the same area together, no geographic isolation. But here is a fish that makes its living scraping algae off of rocks. It's not glamorous, but hey, it gets the job done. The adaptations that make this fish good at getting this food are very special. It needs jaws for scraping. It needs tough teeth for raking up that algae. Its whole body is, and where it's able to, how fast it needs to go. Doesn't need to go very fast because it's just eating algae. Its entire body has adapted to this lifestyle of this food source. But if you look at a different cichlid, like the snail eater, that's a very different type of food. It needs strong jaws to crack open the shells of these snails. So its body is going to be different. Plankton eaters, they don't need jaws at all. They just need to move around constantly and filter out plankton from the water. Fish eaters need to be very fast and agile to hunt down fish that they can eat. And, all these other ones as well. Again, there's no correct answer about how to cichlid. These are all correct answers, and selective pressure is pushing the fish in all of these different directions. So you get a bunch of different fish, all within the same area, because they're all going after different ways of living. And again, these differences, it's not just in their jaws. It could be, in what time of day they're active, and, the size of their eyes to spot the prey that they need, or, being able to spot algae. Don't need to be very bright to spot algae. All these differences will accumulate as they get better and better and better at going after very specific food sources until they are no longer able to interbreed with one another and speciation. Another example of this, real quick, we see finches colonizing islands in the Galapagos. Same sort of story. One ancestral finch came here from the mainland. There is not one correct way to finch. And you get all these different species of finch arising in the same geographic area, going after different food sources. Going after small insects, going after large nuts, going after cactus, going after, some of these are going after stripping bark from trees and grabbing insects underneath there. Lots of different lifestyles and lots of different birds. Sympatric speciation can lead to something called adaptive radiation. So adaptive radiation is, to read from the key terms, speciation where one species radiates to form several other species. And actually, this slide here that said sympatric speciation, this is also an example of adaptive radiation. This was one ancestral finch coming to these islands, and over many, many, many years, selective pressure pushing its descendants in all of these different directions. Again, kind of looks like, spokes on a tire radiating out, this one ancestor having many different descendants that evolved in very different directions. That's what adaptive radiation is. And that happens often enough with sympatric speciation, especially when there are many, many, many different correct answers for how to live. And yet, at the end of the day, despite all the differences and their beak size and what time of day they're active and all that stuff, they're all just still birds. they're all sort of medium small sized birds. And at the end of the day, despite all these differences in these millions of years, they're still very similar fish. They're still very similar ground squirrels. If we want to see big changes, dramatic changes, this is enough to cause speciation. But if you want to see really big, very different species, we got to go back really far in time, because these changes are going to take a very long time to happen. If we want to look back millions and millions of years into the past to see how evolution has played out, we need to start looking at fossils. So the key terms has defined fossil as the preserved remains, impressions, or traces of a dead organism from a past geological age. So remains, obviously bones are remains, and that's what most people think of when they think of fossils. But the definition says remains, impressions, or traces. So yeah, this could be, footprints, or this could be, the impression of a leaf. We've got lots of plant fossils that are just sort of the impressions of the plant leaves that have been preserved. It could be remains. This is what's called a coprolite, also known as fossilized poop, which can give us some important information about organisms. But yeah, all these preserved remains from a very long time ago are very important for looking at stuff and what the world was like and the species that existed a very, very, very long time ago. Importantly, the term fossil record refers to all the fossils that have been discovered to date. So when we talk about the fossil record, we're just looking at all of the information that we have available to us now, all the different, footprints and feather impressions and bones and poop and all the stuff that we've found so far. All that is part of the fossil record. Importantly, this is rare. Fossilization, for something to die and for it to be preserved, for its bones or its remains or whatever to be preserved in a way that could be dug up and analyzed millions of years later, is an extremely rare event. Most organisms that have lived and died on this planet have not been fossilized. Their remains have been broken down by decomposers, other organisms, and there's no trace of them ever existing at all. So the fossil record is cool. We've got a lot of very interesting, important information about, stuff that existed at some point but doesn't exist now. But we do not have everything. We will never have everything, unless we invent a time machine, I guess. But I mean, this is a limited way of looking into the past because it doesn't happen very often. If we're trying to analyze the fossil record and sort of piece something together and get a picture of what the past looked like, this is kind of like putting together a jigsaw puzzle and you don't have all the pieces. Well, again, we'll never have all the pieces. Most things have not been fossilized. That doesn't stop us from doing this. It just means that this is a very challenging exercise to do when you don't have all the pieces. You could still get a good sense of what the picture is. You could still make some connections. But yeah, it's important to understand that this is a rare event. We're not going to capture everything. Now, one of the things that helps us when we're trying to put together this sort of incomplete puzzle is we can figure out how old fossils are. So we know the way, evolution works. It's ancestors, passing their genetic information on to their descendants. And because we can know how old fossils are, that really helps us in sort of ordering what pieces are at the top and what pieces are at the bottom. That helps us put this puzzle together. I won't go into the different ways in which fossils can be dated. Deeper fossils are older than more surface-level fossils. There are more sophisticated ways to measure trapped radioactive isotopes within rocks and fossils to calculate how long ago those things were alive and walking around. But I'm just going to suffice it to say that fossils can be analyzed to know how old they are. That helps us to put them in order. Now, the whole point of looking at these was to connect the past to the present. To look at stuff that, based on these bones and other things that we've dug up, existed a long time ago and connect those things to organisms that are alive today. So here's my statement. Examining fossils of closely related species can reveal evolutionary histories of modern living organisms. And this is kind of the goal. We know what we have today. How did we get here? So I'm going to walk through an example of the evolutionary history of a modern living organism, starting with this thing, artist's recreation based on fossils. This is a very, very distant ancestor to the modern whale. And that's a bold statement because this thing does not look like a whale at all, but evolution is about small changes. So let's walk through this. This Indo-Hyas, obviously walking around on four legs, it is similar to modern toothed whales, like orca whales, based on its jaw, based on its teeth. I won't bore you with the details, but the way these teeth are arranged is pretty unique for a mammal, and it closely resembles that of dolphins and orca whales and modern toothed whales. So that's sort of the initial connection of like, this thing looks kind of whale-like, but obviously it's missing some important whale features. How did we get from here to whale? Well, if you look closely at its bone structure and bone density, you will see that it probably spent a lot of time in the water. Organisms with high bone density like to lurk around in the water and spend time in water. It helps them walk around and not float on the water unintentionally. So we can infer that this thing, could probably swim and spent a lot of time in the water. It's not that weird. There are organisms alive today that have similar bone structure and live a similar life. Now, we can draw a line from Indohyus to Pachycetus. This is a huge difference. This is a massive leap. These things are very different from one another. What you have to always remember, evolution is small. It is about small phenotypic changes, little changes in bone structure or foot structure or skull structure. Indohyus did not directly evolve into Pachycetus. There were many small changes in between these two. But, as I've said, we don't have all those. We don't have all those intermediates. We don't have every single jigsaw puzzle piece in this puzzle. So, again, I'm drawing a line from this to this. There's a lot in between. We just don't have everything that is in between. So why are we drawing a line from this to this? Well, we know that Indohyus is older and Pachycetus is more recent. And despite their differences, there are a lot of similarities, especially in the skull and in the tooth structure. So it's thought that Pachycetus is a descendant of this Indohyus. So what changes have we accumulated now? Well, for one, it looks like it spent even more time in water. Based on its legs, it was probably a very good swimmer, but could absolutely still walk around on land. Another big difference that we see in Pachycetus is the movement of its eyes towards the back, top of its head. Again, we see this in the world today. We see this in alligators and crocodiles. Based on its skull structure, this probably lived the same lifestyle as a crocodile or alligator, an ambush predator that spends a lot of time in the water, mostly submerged, looking to jump out and grab something with its little eyes peeking out of the water. And again, there are enough similarities where we can draw a line from Indohyus to Pachycetus. What comes next? Well, again, in the fossil record, the next thing is something called Ambulocetus. Many small changes between Pachycetus and Ambulocetus. We just don't see all of those. This is just the next one that is available to us in the fossil record. So what's different in Ambulocetus? Well, based on how its limbs are articulated, it looks like it spent almost all of its time in the water, doing a whole lot of swimming, kicking its legs, swimming around, kind of like a modern otter. You also see evidence, paleontologists really look real closely at bones. There's also evidence in the skull of a fat pad, which aids in hearing while underwater. So yeah, this thing probably spent almost all of its time underwater and had pretty terrible mobility on land. And again, we can infer that this is a descendant of Pachycetus based mostly on the tooth structure in its jaw. The next thing that we find in the fossil record is something called Remingtonocetus. This thing was fully aquatic, even worse on land. Based on the isotopes they find in its bones, it lived in salt water, so we're in the ocean now. And based on the bones in its back and its spine, it probably didn't kick its arms and legs like an otter. It probably arched its back like a modern whale. Next in the fossil record, we have Rhodocetus. We're starting to see smaller limbs that look more like flippers. We're starting to see a nostril that's making its way further back in the skull. Spoiler alert, that's going to become a blowhole before too long. And, I'm not going to bore you with all the rest, but we see, flukes start to appear. We see flippers as hind legs are reduced, and the rest. Again, I won't go through every single one of these, but there's a very robust fossil record to get to the modern toothed whale from something that doesn't look like a whale at all. We get there by small changes and looking at the things that are similar from one to the next and tracking these differences as they occur. And again, this takes an incredibly long amount of time. This takes millions and millions of years of small changes accumulating to see these big differences. Here's another example. Just to go away from whales, we could do the same kind of exercise looking at the modern horse. And again, this shows the time scale here. This is like 60 million years to get to this Hyracotherium to, modern horse. Yep, it's getting bigger. The tooth structure is changing. The one I want to focus on is how the limbs change. From walking on its fingers, kind of like a dog or a cat, to bearing all of its weight on a single finger, like a hoof. And as this happens, the side fingers start to grow smaller and smaller as they prove to be useless, as it's, just standing on one toe. You don't need fingers on either side of it. Until we get to the modern horse, which only has the one finger that it's walking on, the hoof. It's lost those other fingers completely. And again, we see these gradual changes by examining this fossil record. As always, tons of individuals in between this one and this one. We're just working with what we've got. We know which ones are older. We know which ones are more modern. We can place together things like this, tracking evolutionary history. And one more real quick, because it's not just mammals, it's not just animals. We can do this stuff with plants too, looking at the impressions of these plants, preserved in rocks. We could put together an evolutionary history for how we got to modern flowering plants from much, much more primitive plants, and, the evolutionary innovations, the sort of things that they built up along the way. All of this is emphasizing this statement. By looking at fossils of closely related species, we can see the evolutionary history of modern living organisms, how we got from this stuff that we dug up to stuff that is alive today. And if we look closely at all of this stuff, this can account for all the diversity of life on Earth. Again, mammals, plants, insects, all this kind of stuff. If you take BISC132 with me, we're going to look at stuff like this, looking at the origin, the evolution of all these different groups of animals. We're going to look at stuff like this, looking at eukaryotic supergroups. Very exciting, I know. We're going to look at stuff like this, which is actually all life on the entire planet, as the You Are Here star with animals. But no, we're going to look at this stuff if you take BISC132, because following these evolutionary histories is something we can do for everything to look at all life on Earth. And I want to close this by emphasizing these are always small changes. This is just microevolution, but taken over a very long period of time. Everything that we talked about in the last recorded lecture earlier within this chapter about allele frequencies changing, it's that same stuff. Adaptations and selective pressure, it's those same things just applied over an incredibly long period of time. We don't have all the fossils. We'll never have all the fossils, but we can put together pictures based on what we have to try to understand how we got to where we are today from stuff that is not around today, but had to have existed at some point because, we've got fossils and hoof prints and poop and stuff like that available for it. So, okay, this is where I close out lecture 4.5. This is the end of the evolution chapter, and this is the cutoff for exam number 4. So exam number 4 is going to end after this. It's going to include all of the evolution chapter. This is the end of 4.5.